this body? What is it?
Fleming of St Mary's Hospital, London, who discovered that it produces the drug known as penicillin.
The question was, which drug would go with which cell?
It's a collaboration that's unexpected because it's between humans and the tiniest organisms.
The howl of a dire wolf.
Dorset lamb may represent a major landmark.
I did not start off planning to design proteins.
Scientific breakthrough will probably be something that we hard to imagine.
Test at birth could map out an individual's entire future risk of disease.
I wanted to get started with kind of your background, right? Like how you got into science. And I read somewhere that you had visited the Galapagos Islands when you were quite young. Is this a true story? What was this experience like?
It is a great origin story, isn't it? It's almost too good to be true.
Yeah.
In my case, it actually is true in that I had amazing parents and they knew that I was really interested in how animals work. I guess you could say the original story was, of course, reading Jurassic Park very young and being somewhat inspired by the very idea. Mm-hmm. But I was always more interested in creating new forms of life and dramatically changing the ones that already exist. And my parents picked up on that and we went to the Galapagos when I was 10 or 11. And that got me fascinated by evolution. So I started reading Darwin and was just enraptured and decided, no, I want to know how to create these. I want to know how to create things as beautiful and wonderful. I want to learn how to make a new tapestry of life and edit the existing one. It was very unwise at the time. I was not thinking at all about the consequences, about the ethics, but I knew that's what I wanted to do with my life, was how to reweave the tapestry of life.
Interesting. And what was the actual trip like? What did you learn? Revelations.
So I learned that tourism is a great way to draw attention to an issue. It is not a great way to protect a sensitive ecosystem. That there's an amazing variety of bizarre species of the Galapagos for all kinds of wild reasons. So there's penguins at the Galapagos Islands, right? Mm-hmm. It's not that far to the equator, but I learned that they're there because the Humboldt Current goes that far north. So why do penguins prefer cold water? Well, the colder the water, fish are ectotherms, so they move more slowly in cold water. So anything that's endothermic has a comparative advantage in cold. So mammalian predators love to be in colder water and have an advantage, and you get more mammals at the top of the food chain.
Interesting.
Penguins are not quite at the top, but nevertheless. So that's why you get a lot of penguins in cold areas, and the Humboldt Current is there. But they coexist with, closer to shore, warmer waters with marine iguanas, which Darwin called the imps of darkness. Right, what a great name.
Okay.
The imps of darkness. And the marine iguanas, of course, compensate by warming themselves in the sun on the rocks, and then they dive in.
Interesting. So two very different species cohabitating. And this kind of spurred your interest in engineering biology. I assume there was many more sort of seminal moments, but eventually you got interested in, actually pursuing this systematically, did a PhD with David Liu, and during this you invented PACE, right? Phage-Assisted Continuous Evolution. This is what made you initially semi-famous celebrity in the science world. Tell us about what is PACE? How did you develop it, and sort of how has it evolved since your career?
Well, my original interest was actually in stem cells and reprogramming for anti-aging purposes.
Okay.
So I went to grad school thinking that was my primary thing, but if I couldn't do that, then I wanted to harness evolution and come up with a way of using evolution to create better tools. So got to grad school, worked in Doug Melton's lab, concluded that stem cells were not going to do anything useful for aging or probably anything else for probably at least a decade.
And you were right.
Turns out that was correct. And I concluded that either way, field didn't need me. It wasn't in need of clever ideas. It was in need of people who could put their nose to the grindstone and really work out some incredibly complicated biology. So I instead arranged to meet with David Liu. And David is very generous and very forgiving, and he loves talking about ideas, which is good, because I went in with a list of a dozen different things that I wanted to do. And pretty much rudely just jumped into it at the very start of the conversation.
Interesting.
But he completely rolled with this, and we just went down the list, and he was like, "Oh, we actually tried that a couple of years ago, and it failed because of this reason. Like, if you can think of a clever way of getting around that, then that would be great, but otherwise, probably not worth it. Oh, this one probably won't work because of this reason. Oh, we've always loved to do that, but could never figure out a good path. Do you have any good ideas for this one?" And one of those was PACE. So PACE is a system for evolving molecular tools. So the basic insight that I had was it's probably more effective to invent a tool that can catalyze the efforts of a lot of other researchers than it is to be a direct researcher working on something important yourself. If that's true, then by the same token, if you can invent a platform for making it easier to invent molecular tools, then you can be further catalytic. You should just go up one meta level whenever possible. Obviously, there are limits, but it didn't seem like that was widely understood as a possible concept. So I thought that there was some neglectedness there. And what I wanted to do was harness evolution as effectively as possible. Evolution is at its strongest when it has many, many generations and when it has very, very large populations. So the question is, how do you harness something that can grow to very, very large populations in the laboratory and evolves very, very quickly, where you can shape the selective conditions to favor the evolution of a molecular tool of your choice? So I looked at what are all of the different ways that you can cultivate something in the lab that replicates very quickly. And the fastest is a bacteriophage, virus that infects bacteria. You can grow these to populations of 10 to the 10th, so easily 10 billion. But usually it's more like 100 million cells per milliliter or so of culture. And then each of these can be infected, and that sort of counts as one. And the fact that it's spitting out a ton more phage than that doesn't really count, because what you care about is what is the basal population that gets to replicate. It's a lot of extra technical detail, I recognize. But point is, they replicate as quickly as every 10 minutes. Usually it's more like 30. So my idea was, how do you engineer either the phage or the cell to ensure that the phage will optimize a molecular tool that you encode on it? And the way I thought we could do it, I just proposed this to David, as I said, there's got to be some way where we can take a gene from the phage that it needs, where production of this, whatever protein it encodes, is proportional to the phage's ability to replicate. Move that gene into the host cell, and then program the host cell to produce it in proportion to the molecular activity that we want to see. 6 years later, we had it working reasonably robustly.
Got it. That was a better explanation than anything I'd read online, actually, which is great. You sort of did my job for me. But the protein that you're talking about here that was necessary for its reproduction is P3. And you moved it into E. coli as opposed to it used to be in the phage genome. And you have this selection plasmid, and then you also have this like mutating plasmid, essentially. Talk to us about a little bit about the actual mechanics of that and what went into the engineering of this system.
So the initial thing that, for anyone who is currently doing research or thinking of doing a career in research, that I will emphasize is, if you don't have expertise in a field and you think you're going to need that expertise, then you should sacrifice weeks or months of your time to go and train with someone who is actually an expert. Because if I had done that in terms of phage biology, I would have saved myself a lot of time, especially if it was in the biology of that particular kind of phage. And I didn't, so a lot of it was figuring out which genes can you mess with, which genes can you move, and how do you actually grow phage effectively and robustly. Once I worked out all of that, then the question was, how do you introduce diversity? Because it's not just about how many different evolving members of your population can you have. Ideally, you want every one of them, more or less, not literally everyone, but close to every one of them, to have a different mutation or set of mutations. And natively, the polymerases of most organisms are accurate enough that you don't get a new mutation in every copy or anywhere close to it.
At least for genomes that small. Right?
And the reason is, even for genomes that small, so the larger the genome, generally the more accurate the polymerase, because the less tolerant it is of mutations. So the more essential genes you have in a genome, then the greater the selection pressure for accurate replication. Because if you make a mistake in an essential gene that inactivates it, then that progeny is not viable, and natural selection will act against you. The problem with increasing the mutation rate a lot in a bacterium is that it will accumulate mutations in essential genes, and it will die. So the trick with PACE that we took advantage of is that we're not trying to evolve the bacterial cell. It's okay if the bacterial cell takes a ton of damage and dies. In fact, we want it to take a ton of damage and die, because if it doesn't, if it can replicate as well, then whatever we're selecting for, whatever activity it is, you could end up with something in the cell that interferes with or contributes to that activity, when really we want the tool encoded on the phage to evolve.
Got it.
Everything in the cellular genome is a potential cheater that could mess with our selection. And the number one saying in directed evolution, which applies very much to modern AI as well, anything where you're growing or training rather than directly engineering through rational design, is that you get what you select for, which is not necessarily what you want. So we wanted a simplified system where the only thing replicating was the phage. We wanted none of the cells to be able to replicate. So not only did we not care how much mutational damage the cells took, we actually wanted to ensure that the cells never got to replicate. But there's a much easier way to do that, which is you flow the cells through the chamber faster than the average bacterial division time, but not faster than the typical phage replication time. So new cells come in, they get infected, they spit out new phages, and they go out faster than on average they can divide. So that was ultimately the key to PACE, was engineering the cell to replicate the phage in proportion to the activity that we wanted, while increasing the mutation rate to a much, much higher level than normal. So this is a really complicated system, right? This is creating effectively a synthetic ecosystem of an engineered host cell, an engineered bacteriophage, changing the mutation rate, changing the selective conditions on that bacteriophage, and then engineering the phage genome by moving a gene around and encoding a desired molecular tool in its place.
Yes.
That's why this took 6 years.
I see.
I do not advise my students to do anything like this because there was no backup plan. This was a purely engineering project, purely directed evolution, platform development. There was nothing that we were going to discover in the process of building it that would be publishable. So it's a good thing it worked.
Yes, and in just the time frame that it was pretty, you were cutting it close there with 6 years, 7 years, 8 years. I think like, especially recently, there's pressure to graduate sooner. But at least at the time, it looks like you had a few more years where you could have failed and still succeeded. But just to sort of summarize this, you've changed the selection plasmid with these low fidelity polymerases that basically introduce mutations at a higher rate than normal, and then you're flowing these E. coli cells through, constantly replenishing the E. coli pool, and these low fidelity polymerases are introducing mutations in the bacteriophage gene that codes for the protein that you're trying to optimize. Anyway, it's a very complicated system. I'd encourage our listeners to read the original paper or to sort of watch a video of you explaining it with slides. But essentially, you created like a Darwinian engine, right? Like it performs real-world computation. It figures out how to optimize for some sort of like biochemical characteristic of this enzyme. Obviously, you have to do a lot to link the biochemical activity of the enzyme with the selection pressure of the phage, and that's a complicated thing to do. But like once it's done, you have this Darwinian engine that you can use to optimize the activity of any enzyme, and potentially for industrial applications or for others. Talk to us a bit about if there's anything you want to add to what I just said, and also what are the uses of this technology for biology, for humanity? Why should our audience care?
Yeah, so broadly speaking, I would assert that progress in biotechnology is primarily driven by our access to tools with which to engineer it. And the most obvious example to point to is CRISPR. Now, CRISPR is, as most listeners probably know, a pair of molecular scissors that you can use a guide RNA to direct to a corresponding DNA target, and natively CRISPR cuts the target. Cells respond to a cut DNA by trying to repair the damage. If you give a similar looking sequence fragment, it will sometimes stitch in your fragment in place of the original. This has revolutionized genome editing. But David's lab subsequently did one better. They turned CRISPR into a tool that doesn't cut the DNA, or at least doesn't cut both strands, but instead makes very precise changes of just one base or several bases without having to cut the DNA. And this is important because if you want a gene therapy, if you want to change the DNA in a patient, cutting both strands is potentially really bad because the cell might incorporate your new fix. Or maybe you want to just disrupt a gene, in which case it will usually do that. But there's always the risk that that broken end is going to be repaired by jamming it into some other piece of DNA. And if it jams into the wrong piece of DNA, then you could turn on an oncogene and potentially give the patient cancer, which is always the great fear of gene therapy. Okay, I guess you activate the immune system the wrong way and the patient dies immediately is even worse. But giving them cancer is pretty bad.
Yeah.
So the folks in David's lab had this really clever idea that you could instead not cut both strands and instead attach to CRISPR an enzyme that would chemically change one base into another, such that when the DNA was replicated, you would end up with a different base in that place. And that this then would be a safer way of accomplishing gene therapy in people. And this is what led to first base editing, which is just changing one base into another. And they've done a lot of variants of that. And then eventually prime editing, which is where you attach the sequence you want to insert to the guide RNA, along with an enzyme to turn that RNA into DNA that then gets inserted into the site being edited by CRISPR. And the way they got both of these working is they developed a clever selection using PACE, in which they put the CRISPR with the modified additions onto the phage, and then they engineered the host cells to select for the desired mutation or insertion of new DNA. And then they optimized it until it was extremely effective at making that particular change, orders of magnitude better than what they started with. And it optimized it using mutations that we could not have predicted would improve it. Because the reality is that even with modern machine learning-based biodesign tools, we're still not that great at predicting, in particular, enzymatic activity, which mutations are going to make a biomolecule that is complicated, that works by stabilizing the transition state of a reaction, in particular, more effective at its job. More specific, more discriminating, you name it. And so the best way to do this is still generally to make a bunch of versions, throw them all at the wall, see what sticks. And then do that again and again and again. The best thing about the biodesign tools is they will now often give you something to start with. That is, there might not be any natural enzyme that has the right characteristics. And now, increasingly, we can design that. But you still typically need to optimize it. It's very seldom, even with the Baker lab and others at the forefront of the field, still typically don't get something that's amazing on the first try, except for binders. But binding is relatively simple, just strict molecular interaction. It's not stabilizing a transition state, which is much more of a high finesse operation. So PACE was important to optimizing this, and tremendous credit to folks in David's lab. That was a lot of optimization required to get such a large and complicated enzyme to work. For some of them, they had to split CRISPR into 2 pieces and use an intine to splice them together, producing 1/2 from the host cell and 1/2 on the phage, because the phage really wasn't happy with a 7,000 base insertion.
Yes. Yeah, this is something that really only David is very good at, and maybe a few others. So I think I'm understanding your premise that tool development is incredibly important. It kind of unlocks branches of medicine. There was a drug, I guess you could call it a drug, Casagavir, that was approved that used base editing in patients for sickle cell disease. So clearly there are portions of the tech tree that are being unlocked by tool development and by PACE. And what I'm also hearing from you is that these kind of protein design tools, these in silico methods, just don't have the requisite data set sizes to be very good at doing sort of one-shot design and optimization. And I think one thing I was going to ask was, like, how far off are we from this? Like, how many more years is it going to take until we have in silico protein design that is as good as real-world computation through PACE?
In a lot of ways, it's always, excuse me, in a lot of ways, it's already better. And when I say that, it's because the in silico tools can take data sets generated in mammalian cells or human cells on, say, histone-wrapped mammalian DNA, if that's what you want to alter. It's relevant to the system that you want. Whereas the downside of PACE is you're doing this in bacteria. And sure, you're throwing a ton of evolutionary optimization at it. Like, base editing just got a lot better. But you still need to optimize it in the situation of interest. So that's a major, major advantage for design. Also, you don't need to have any starting activity. Sometimes you can design something that has an initial activity that doesn't exist in nature. So it's a lot broader in utility. But when will we be able to design enzymes reliably? Again, the Baker Lab has managed to do it for a couple of cases. Doesn't seem super general. In all cases, they knew what the transition state they were trying to stabilize was. And Francis Arnold's lab and some others have managed to come up with some novel chemistry that doesn't exist in nature in this way. But then they still have to optimize it after the fact. The design tools aren't good at getting anything near natural catalytic efficiency. And as you alluded to, the problem seems to be data. So what Vikram Sundar in my group has found is that it looks a lot like these tools are still data limited. And we know this because we've been looking at, in particular, engineering proteases to cut arbitrary targets. So Bo2 in the group developed a very clever method of trying to gather tons of data, because we reasoned if data is in fact the limiting factor for machine learning and biology, for biodesign tools, then we needed a way to collect as large a data set as possible. And in principle, you can kind of extract this from PACE, and some folks in David's lab have tried to do this. So PACE, it's doing a really good job of trying to optimize along whatever is accessible to it. So in principle, you can have some starting point. You can run a bunch of PACE experiments in parallel, ideally, and you can track to see which mutations it comes up with. And you can also notice which ones it presumably could access, but didn't actually take over. So in principle, with some level of confidence, you can say whatever it spat out outcompeted everything that was accessible that didn't win. And you can use that to infer what has less fitness than the thing that actually won. And that is relevant data that you can feed to machine learning by inferring fitness. But it's honestly not great for this. It wasn't optimized for that purpose. And although my group has also developed a robotic implementation, others have as well. Ours is probably the highest throughput because it uses a liquid handler. So you can run dozens of parallel PACE experiments, each in different deep wells of a 96 well plate on a liquid handler, where it just does all of add the new host cells, remove the excess host cells, add any inducers that you want. And it gives you very fine control. Because any given PACE experiment, what should your ideal mutagenesis rate be? How strong should your selectin pressure be? Do you want really strong selection early? Do you want weaker selection early? Do you want intermittent selection? there's a lot we don't know about how to optimize molecular evolution. So we built this robotic system to let us change the parameters. So Erica De Benedictis did a lot of this work with Emma Corey, and they both run their own groups now. And really, Emma's leading development of this Prance system.
I've seen the video of it. It's quite insane.
Yeah.
It scares me for the future of wet lab biology.
Well, we actually learned a lot in that because it turns out that a liquid handler, to our dismay when we had this, clever idea, Erica's face when she first had to work with the software that came with the liquid handling robot was priceless. Shall we say that these companies are very good at making hardware, and their software leaves something to be desired. In particular, because they have to build software that works for biologists who are not, shall we say, necessarily that skilled at working with programs.
Understood.
Pretty much always works with GUIs. You do not have a command line at all. Most parameters that you might want to change are not parameters that you can change because they assume that you are using this liquid handler for essentially high throughput drug screening. And that is it. If you want to pipette different amounts from different wells at the same time because you're trying to get a lot done on the deck in a limited time frame, the hardware has the capability to do that. The software just would not let you. So Erica worked with Dana, another student in the lab, to develop what we called PyHamilton, which let you program a Hamilton robot using Python rather than the native system. And later on, Rick Wieringa and Stefan Golas developed PyLab Robot, which extended this to other manufacturers and talks directly to the robot at the firmware level rather than going through the layer of the manufacturer software.
Got it.
So you might say, in order to get PACE working into Prance on a liquid handler, we had to write new open source software to make liquid handler control much easier and more accessible to everyone. So if you use a liquid handler, I highly recommend checking out PyLab Robot. It is just way more flexible than pretty much any manufacturer software that's out there. I guess OpenTron's the only one that arguably comes close, but unfortunately, OpenTrons are not as versatile as any of the more expensive robots. And so if you really want to unlock the potential of lab automation, you need good software and good hardware together. And I'm really excited where OpenTrons is going, but frankly, a Hamilton is just much more effective.
What are the opportunities, you think, in the space of lab automation that are maybe like low-hanging fruit and deserve to be exploited? Let's touch on that before we get to meat sacks and how much time humans have left.
So I think the future of biology is probably mostly automated in the sense that machine learning tools are definitely better than we are. That is, I expect them, they're already better, obviously, at predicting protein folding, say, than we are. And they're going to be better at predicting protein design than we are. Arguably, they already are. And eventually, they're going to be better than directed evolution at creating useful new molecules that do what we want. But in order to get there, we're going to need a lot of data. So as I was alluding to, Vikram found that when we... Actually, I never got there. So Bo developed this system to gather a ton of data for machine learning by measuring individual activities of different molecular variants of a given protein that does some work in a cell. And Bo's clever idea was to dispense with all of the excess robotics or complicated machines, or especially inference. That is, if you have to measure the activity of your whatever it is you're interested in by how bright the GFP is in a cell, or especially how tightly it sticks to something, and then you look at relative enrichment. The problem with the how bright is the GFP is you end up having to have your fax machine sort cells by brightness into different bins, and then you look at the relative enrichment in different bins to infer how active it is. And this is inherently very noisy. So Bo's insight was there's got to be some way to do it without having to have a complicated flow sorter. And he reasoned, well, what if we took a base editor? And instead of taking a paste circuit, which produces this phage protein 3 in proportion to the activity of interest, what if we had it make a base editor in proportion to the activity of interest? And then we put target sites for that base editor next to the molecular tool that we're testing. So envision there's 60 plus recognition sites for this guide RNA that have a bunch of cytosines, and this base editor turns C's to T's. So there's a ton of sites, and they're all right next to the gene that you're trying to measure the activity of. So you make 1 million variants of this gene, make that library, you clone it into your construct that has a ton of base editor sites in cells that will produce base editor proportional to the activity. Really active copies will produce a lot of base editor in their cell, and they'll start mutating those sites. So after a given amount of time, you have a gradient in terms of the number of mutations. Cells with more active copies will have more mutations. Cells with less active copies will have fewer mutations. And the neat bit is you can play with the timing. If you want to know which ones are absolutely most active, then pull them all out and sequence them very early. And then you'll be able to distinguish the really active ones from the only pretty active ones. And if you want data on ones that can differentiate between just a little bit of activity and a little bit more than that activity, then you can run it for a long time. The really active ones will all saturate, but you'll be able to zoom in on the difference. So by playing with this time element, you can actually get much more detailed data. And it's a direct measurement. But it's still noisy. So here's a tool, we call it DARMA, because you have to come up with a cool acronym for everything in synthetic biology. That's just how it is, right?
Sure.
And DARMA gives you this ability to measure for 1 million different variants at once. It's basically how much are you willing to throw at nanopore sequencing costs. That is how many data points you can get. And Bo's developed ways of enriching for more active ones so that you don't waste your sequencing on less active data points, which are less useful for training the models. You want some data on what doesn't work. That is useful data. But in any given randomly generated library, most of them are going to be deleterious, more than you'd want. So you usually want to combine a selection for the more active ones that is not perfect in order to enrich for the more active ones and then get some data on what doesn't work. And that seems to help the machine learning the most. But it's still noisy. And to our shock and horror, the state of the art for training machine learning models on biologically collected data involves throwing away the error bars, taking the mean or the median. That is the fitness that you throw in during training.
Got it. What would the alternative be?
So ideally, you want to work in an actual uncertainty value. But what is that uncertainty? What should it be? You can't really just get it out of the measurement. You need to measure it in the first place. But then you also need to produce what's called a probabilistic fitness landscape. You need to train on that probabilistic fitness landscape, which means you need to convert your actual data into this landscape, which is a machine learning problem in and of itself. So Vikram developed this method called flighted, which essentially solves this problem. And it's nice because you train the flighted model for whatever selection you're using. So you can use DARMA, and then you can use, say, phage display and enrichment or FACS. Once you've trained one flighted model, you can use it for any experiment of that type thereafter.
Got it. Well, let's take one step back, just because we're getting very, very technical here. But to sort of summarize, you're using mutational burden generated by these base editors as a proxy for the activity of the enzymes that you're trying to collect data for, right? And you can do this in a pooled way. You generate like a large library of millions of these variants, and you can change the amount of time that you let this run in order to increase or decrease the mutational burden, and that allows you to basically, in a more high fidelity way, ascertain the difference between activity levels, if the activity levels are kind of similar to each other, right? And this allows you to generate a lot of data, and you're hoping that these techniques will enable us to turn PACE from biological, like real-world computation into in silico computation. Is that a good summary?
Yes, although this doesn't actually have anything to do with PACE. It's just a totally different method.
Okay, got it.
Dharma is purely about measuring as many different variants as you can create to gather as much data for machine learning as possible.
Yes.
And so Bo's goal, for example, is to gather lots of data on which proteases will cut which particular sequences and will not cut other sequences. Because ideally, certainly for a therapeutic, you want a protease that's highly specific. Will cut only the sequence that you want that's therapeutically relevant and is not going to go around chewing through all of the other proteins in the body.
Okay.
So how do you generate that? We don't have nearly enough data on the fitness or activity of different proteases across different substrates. So Dharma is a way where you can change the substrate that leads to production of the base editor. Use ideally a library of different substrates and then a library of different proteases, and then that will generate enough data such that eventually we hope to be able to have a machine learning model where you say, I would like a protease that cuts this particular sequence or this set of sequences and does not cut any of these others that are most similar to them that are found in whatever system we care about.
Understood. Okay. And I think this is just like a very interesting time to transition into just the confluence between these like large language models and these like biological machine learning models and what the intersection of them is going to be, right? Obviously, you're very afraid of these LLM systems being exploited by bad actors to create engineered viruses, and I imagine that some of your time spent developing these tools maybe has led you to regret making them so widely available, right? We have PACE, we had the concept of gene drives that you basically proposed in George Church's lab at the Wyss Institute, and I imagine this opened your eyes to the sense that, oh wait, this could be used for nefarious purposes, right?
Yeah, I'm not actually all that worried about PACE, and I'm not really worried about Dharma or even immediate applications to molecular tools. Gene drive was the one that, we proposed it, we built the first one, it worked, but the first one that we built actually was to knock out a particular gene in yeast, and the Ura gene. But when we built that first one, we also built another gene drive that would target the first one and restore the gene. And this is important because I only disclosed that CRISPR-based gene drive was possible, and I should probably explain what that is if any listeners haven't heard of it. So the idea of CRISPR, of course, is you can cut a target gene and copy in a new sequence of your choice. Existing life does this all the time. There are already nucleases in existing organisms that reproduce sexually that cut the part of a chromosome that doesn't contain them, thereby forcing the cell to copy over the gene that encodes them. So you can imagine that this is very advantageous because it converts chromosomes that don't have this nuclease gene into one that does have it. So in an organism that reproduces sexually, that exchanges chromosomes, then any nuclease gene that has this trait can actually copy itself onto a chromosome that previously lacked it. It doesn't have to outcompete it. It doesn't have to provide any fitness benefit to the cell. It just has to cheat at inheritance by directly copying itself. So the most famous one is the ISCI1 endonuclease in yeast, which actually is one of the more common, ultra rare cutter endonuclease enzymes that we use in the lab. It works this way. There are many others. But what I realized was, actually coming from a completely different angle, I was wondering, how readily will we be able to edit wild organisms? Because my original Galapagos vision was, we don't just want to tinker with things in the lab, we want to tinker with things in the real world. Ultimately, it's not just about being to create creatures as beautiful and wondrous and awe-inspiring as those we see all around us. It's about learning to do so, frankly, better. Because nature is amoral. It has no care for suffering. And once you acquire a new technology that lets you address a problem in the world, if you use it and something goes wrong, that's totally on you. But if you choose not to use it, then you are morally responsible for the continued existence of the problem. So once we learn to rewrite the tapestry of life, if we use it and screw it up, that's on us. But if we don't use it, then we are responsible for all of the suffering we could have averted. And so I wanted to know, could you edit wild organisms with CRISPR? And I was thinking, well, obviously you could, and you could release them. But in general, when you edit an organism to do something useful to us, you're redirecting its resources to doing that thing instead of to replication. So natural selection is almost always going to select against your engineered trait. Mm-hmm. But then I thought, well, but wait a minute. What if you encode CRISPR along with the trait that you want, with the gene or genes that you want to insert? Encode CRISPR right next to it. Then you're not just engineering an organism with your desired trait. You're engineering an organism with your desired trait and the machinery to perform genome editing to insert that trait. So I thought, if you did this in an organism that was sexually reproducing, and any offspring that inherits one copy of the CRISPR system and the trait, and one wild copy, if you program CRISPR to only turn on in the reproductive cells, the sperm and egg producing cells, then it will turn on, it will cut the chromosome that lacks it in the heterozygote that has one copy of each. And then that cell will repair the damage some fraction of the time by copying over your desired genes and the CRISPR system. Mm-hmm. So then you've gone from a reproductive cell that has one copy of your engineered trait to both of the copies having your engineered trait and the CRISPR system. So if copying is perfect, you've just doubled the fitness of that gene in heterozygotes. That is a huge fitness advantage, doubling in heterozygotes. Yes. It's enough to swamp almost, not any, but almost any effect on the replicative fitness of the host organism. So what that means is you have something that is fitness positive in the wild, if you can make that work. So fitness positive that you can spread things that are even harmful to the population's reproductive fitness. Mm-hmm. In the extreme, imagine that you spread the trait of being male. What would happen to that population?
Fascinating.
And there's ways of doing that same suppression trick. What if you disrupt a gene required for female fertility that is recessive? Then any female that has one copy of the gene drive is perfectly fertile, but all of her offspring are going to inherit a gene drive, therefore broken copy. So if you have one copy, you're fine, but any females born with two copies of the gene drive are sterile. So the drive will spread very rapidly when rare, and then as it becomes increasingly common, you get more and more and more sterile females born, and it will crash. So Austin Burt, very clever biologist at Imperial College London, first outlined that this could be done with gene drive way back in 2003. And there are a lot of folks working with him who tried to optimize homing endonucleases to do this. The problem is a homing endonuclease cuts one site. It's a real pain to engineer it to cut other sites. And what do you imagine happens if the cell jams the broken ends together and creates a mutation rather than copying your gene drive? The answer is it's just created a gene drive resistant allele that can't be cut and doesn't encode the gene drive. So if the drive is costly, that broken allele is going to outcompete the gene drive. So in order to make a gene drive that's evolutionarily stable, you need the ability to target a gene that's plausibly important for fitness at multiple sites, such that if it acquires mutations, that's harmful. But if it does the correct copying of the gene drive, then it gets to keep its important gene intact. And it has the gene drive. So that's the way to do evolutionarily stable. So because of CRISPR, we can actually multiplex. We can target multiple sites using a gene drive. And we can target any gene we want, including costly ones. And that opens up the general principle of population editing in the wild.
Fascinating.
So then we have to talk, what about offense, defense, balance?
Yeah, we should definitely get to that. But just to take a step back, I mean, there's positive applications of this kind of technology. I don't think it's being used in the screwworm fly, right? They're using a different radiation technique, I think, to make their populations infertile. But presumably it could be used to dampen down pest populations in a population stable manner, as you mentioned. Tell us about what's the good that can come from this besides what I just mentioned.
So there's at least 4 species where you would probably want to use the full power version of this. So first, let me be very clear. You make something that's fitness positive, you release it in the wild, it's going to take off through every population connected by gene flow. Mm-hmm. You're not going to be able to stop it. And we know this from studies of invasive species ecology. You introduce the invasive species, in general, you're not going to get rid of it. There's some outside exceptions where on very small islands, we have managed to get rid of rats by literally carpeting the entire island with essentially bait with rodenticide. This is not always great for other species. If you do it thoroughly enough and repeatedly enough, then you can in fact get rid of the rats. But this only works in small islands. Mm-hmm. Everywhere else, invasive species comes in, that's it. Pretty much the best you can do is biocontrol, introduce some specific predator of that species, and then that will reduce its comparative fitness and therefore abundance so it doesn't cause as much harm. But you're never going to get rid of it. Same with the gene drive in the sense that once it's there, it's going to spread to every ecosystem that it can invade where it has positive fitness, which in this case is if you have any gene flow between 2 populations, the gene drive will invade. Mm-hmm. And that means that if you're going to use this against a species, you're probably going to use it against the species, all of them. And you might say, oh, well, we're only going to hit rats on this one island. And you'd say, wait a minute, how did the rats get to that island in the first place? Did humans deliberately bring those rats? In the case of the black and brown rats that are a problem, the answer is no, that was very much not deliberate. Rats are just great at hitchhiking. And if the originals hitchhiked, the gene drive ones will hitchhike too.
Mm-hmm. And presumably because these binding sites are shared between other species, or is there another cause of the transfer?
Yeah, so that's a great question. Can you accidentally affect a non-target species just because it makes fertile hybrids? So basically, if there is gene flow between them, then you could plausibly hit an off-target species. And this is reason to target sites that are not potentially conserved across species that you wouldn't want to hit. But if the rule is if you do this full power, and there are ways of limiting it, and a lot of what my group did subsequently was try to come up with ways of localizing the effects. Because ideally, you would want to let every household, ideally, or at least every neighborhood, decide, do they want to edit the local wild organism population or not? Let everyone make their own decision. But full power version, it's the whole species. So which species do we want to hit the entire species? Has to be exclusively sexually reproducing. Has to have a reasonably fast generation time. Rule of thumb is faster than 2 years, preferably faster than 1 year. And you want to hit the whole species. Number 1, malarial mosquitoes. Malaria kills 600,000 plus people a year. Usually about 500,000 of those are children. In the course of this conversation, a couple dozen kids have died of malaria. Sure, it would be great to do something about that. It's most cost-effective, among the most cost-effective life-saving donations you can make is for bed nets, because malaria is just that bad. But we're not going to get rid of it with bed nets. But if you engineer the mosquitoes to crash their population to a level too low for transmission, then in concert with other interventions to suppress malaria, we could plausibly get rid of it. You don't have to get rid of the mosquitoes. There's 1,000 plus mosquito species in Africa, for example. You're just hitting the ones that transmit malaria. They don't seem to be ecologically important. Predators don't care which mosquito species they eat. There's nothing that specializes particularly in those ones, as best anyone can tell. If my children were at risk of malaria in Africa, I would say do it. Don't bother safety studies, like the overwhelming moral imperative of doing something about malaria would be overriding. So that's how I would vote. But I don't live there, and my children aren't at risk, so I don't actually get a vote. But that would be my recommendation. Similarly, same sort of areas, schistosomiasis. Horrific parasite, causes all kinds of complications, but in particular, growth stunting in children. And there's 250 million infected people. Costs pennies to cure them, but it's so omnipresent that you still have 250 million infected people, even though it costs pennies to cure. And if you stay infected a decent amount of the time, then you get growth stunting, and in a decent fraction of children, you probably get, at least the evidence suggests, you get cognitive stunting too. So it's not as nearly as bad as malaria in terms of causing deaths, but in terms of causing loss of human potential, it might be even worse.
I see.
They're sexually reproducing. You could hit them directly. Then there's God's 8th biblical plague.
Which one?
This is the one where you can call me hubristic.
Okay.
But if you think back to all of the plagues of Egypt, I believe number 8 was locusts.
Oh, yes.
Plagues of locusts, which obviously, given that they're in the Bible, have been plaguing us for quite some time. But most of the time, the desert locust is a harmless desert grasshopper. It's actually solitary. In one of those amazing transformations in all of biology, when it rains a lot in the desert, then of course the desert blooms. And these desert solitary grasshoppers start eating that vegetation. And they eat it all up until it's gone. It's the desert. But then there's a lot of grasshoppers. And their proximity, some combination of chemical cues and actually physical contact between them, causes an epigenetic change. And it's epigenetics actually inherited by their offspring. They change color and they become gregarious. So they form these gigantic swarms and fly out of the desert and eat everything in sight.
Interesting.
So a locust swarm, a large one, will eat roughly as much as the population of, say, Paris every day.
Wow.
So you can imagine what this does to a vulnerable country's agriculture. But the thing about it is, where they go is totally dependent on the weather. Where they arise depends on the vagaries of the weather. Where they fly depends on the vagaries of the weather. No ecosystem gets hit with a locust swarm on a regular basis. And we know from population genetics that the solitary desert locusts don't have to become swarms to persist. Which is to say, if you use the gene drive to break the transition from solitary to gregarious, and we actually know some good genetic candidates for this, you could turn off swarming in the desert locust. You could tame God's 8th biblical plague, and they would just remain harmless desert grasshoppers until we decided that we were no longer vulnerable and could switch them back.
Fascinating. Yeah, it's almost like full circle for humanity on that front. And then I presume the last species is the screwworm?
The last species is indeed the screwworm.
Okay.
So here is the one where you should care most about this one if you care particularly about animal well-being. So New World screwworm is not for the sensitive of stomach. This is a botfly. It lays its eggs in open wounds, anything as small as a tick bite. And it's called the screwworm because the larvae are screw-shaped. And unlike most maggots, they devour living flesh. They screw their way in. It's horrifically painful. And then once they've eaten enough, then they fall off and metamorphose into flies, mate. But the ones in the wound cultivate bacteria that emit volatiles that attract new gravid females that lay more eggs. So you get this macabre dance in which the screwworm, generation after generation, devours the animal alive.
Yes.
And my rough back of the envelope, it's very rough, it's the back of the envelope, suggests that there's perhaps 1 billion or so mammals and birds, only warm-blooded things, that's what it preys upon, affected at any given time. Doesn't mean they're all going to die, doesn't mean they all have the sustained infestations that are actually going to kill them. Only exists in the Americas, and a couple of years ago I could have said only exists in South America because we got rid of it in North America by irradiation sterilizing screwworm larvae and releasing tens of millions of them across the landscape.
Yes.
So that the sterile irradiated ones would mate with the wild ones. They only mate once. This suppresses their reproduction if you release enough of the sterile ones. And so we released 500 million a week, pushing them west across the United States, out of Texas, and then down through Mexico to the Isthmus of Panama, where we maintained a living wall of typically it was every week about 10 million across Panama to keep them out.
Yes.
But recently they escaped and snuck through, and now they're all the way back up into Mexico. And since we know that the ecological niche still exists, they're not going to go away unless we get rid of them. And South America is even better for them ecologically. So you're never going to get rid of them through irradiation, sterilization, and release. Too expensive. But a gene drive.
Yeah.
So why is this a moral imperative? Well, obviously it's a tremendous amount of animal suffering. Is it worse to be a factory farm chicken or to be devoured alive by flesh-eating maggots? I don't know, but if you assume we're going to factory farm for another 100 years, then we know that there's about, let's say, 100 billion factory farm chickens a year. That'd be a little more than we do now. Let's assume we do it in a little bit worse. For 100 years, that's 10 to the 13th broiler hens. But a billion or so host organisms devoured alive by screwworm every year. Typical lifetime of an insect species is several million years. So a billion times a million is 10 to the 15th in expectation if we don't do anything. So is it 100 times worse to be a factory farmed broiler hen than to be devoured alive by flesh-eating maggots? I don't know, but seems unlikely. But given those premises, if you're willing to integrate over the long term, it might be more important for animal well-being to eliminate the New World screwworm from the wild than it would be to end factory farming tomorrow rather than in a century.
I hear you. Wouldn't the pushback argument on that front be that if we do this anytime in the next 100 years, it's still okay?
Sorry, that it's still okay in the sense that...
As in, maybe not okay is the right word, but if we're integrating it over a very long period of time, then sort of the time frame that we introduce gene drives is not
negligible.
Important.
Yes, that's absolutely correct. Yeah, it doesn't really matter.
Okay.
I'm just worried, are we going to be around long enough to
I hear you.
do
I
it?
see.
And if we are stable in the long term, then of course we're going to do it, and probably much more, I would hope, in order to make the world a more ethical place.
Yes. So just to take a step back here, I mean, there's a good Kaz video out there on the screwworm fly that I encourage our listeners to watch. But essentially, we're maintaining a living wall of infertile screwworm flies that we're mass producing, and this is, I guess, we're taking advantage of the fact that they reproduce only once in their lifetime, and we're compensating for the selection pressure by sheer numbers of these flies that we're releasing into the wild. And with a gene drive, you're solving for that selection pressure problem in a completely different way that does not require a large number of these organisms. It can be just a few. It could be 100. Maybe you can correct that number. And so we have a moral imperative to sort of, at some point, get rid of these species and use gene drives for good. The problem, though, is like if we do make a mistake, one, it might set us back in terms of like public perception, and it could be sort of worse than anything. Might be that. I mean, I've been reading The Children of Time recently. I'm not sure if you've read that. Mm-hmm. It's very much just a topical book for this conversation, and like these gene drives go quite bad on this other planet, or maybe good for the spiders, but bad for humans. Tell us about like what are the risks here? What should we be worried about and cautious about?
First of all, we don't understand ecosystems. So doing ecological engineering is an inherently daunting task. But I might point out, as our listeners are doubtless aware of, we don't understand the human body and how it works either. Like most of our existing approved drugs, we don't actually know with great confidence exactly how they work. We are confident for most of them they do work, though. And somehow we manage to make and approve drugs despite not always knowing how they work, or figuring out only much after approval. And the equivalent for ecology of those clinical trials that allow us to determine do they work and are they safe is a field trial. That is, you make the same change in a small, ideally localized, ideally isolated population in a similar ecosystem, and then you watch and see if anything goes wrong. Doesn't matter to you what specifically goes wrong. Maybe it's going to be something wrong with a predator. Maybe you're releasing pressure on invasive species that are already there or something. There's a lot of things that could go wrong. The point is, we don't understand the system. We can't even articulate all of the different things that might go wrong. But if we make the change that we're planning to make at scale in this small environment and watch it and just look at everything, if nothing obviously goes wrong, then it's probably fine. Just like you give the patients the drug in the trial. If they're fine, then okay, you're probably good. Yes, in principle, you could have broken something that's only going to manifest in them years later. But we know by now that that's really unlikely. You kind of have to do that deliberately, in fact. Same as far as we can tell, probably true in ecology. And in some ecosystems, you can, of course, look at things in advance to try to be able to predict it. I wouldn't say with confidence, but mosquitoes, as I mentioned, the big argument against using gene drive against malaria is maybe the ecological consequences would be worse. But everything that anyone has come up with, including people who frankly don't want gene drive to happen because it would be a big visible moral victory for biotech. And they hate biotech because, not because they're actually anti-biotech, but because big ag uses biotech. And so they are trying to smear big ag by its use of biotech.
What's big ag, sorry?
Big agriculture.
Agriculture.
The Monsantos
Got
of
it.
the world. Obviously, it's been bought more recently and does not go by that name any longer. But there's a lot of folk who don't like the way we do agriculture, don't like big companies dominating agriculture. They want small farmer collectives to be the way we do agriculture. And they want to support European agriculture over large-scale American corporate agriculture. And so they chose the difference between them being that American agriculture uses biotech. So they figured, well, you demonize biotech, then you push back against large-scale corporate oligopolies.
Interesting.
And this is what Friends of the Earth and their ilk do. It's not that they want the children to die. It's that if we prevent the children from dying using biotech, then it will be a lot harder to demonize GMOs. Because here we just took the most, I guess you could say, powerful form of biotech intervention at the whole population level and edited not just things that we eat, but an entire wild species. And if that goes well and we save a lot of children, that's a great moral victory.
I see. Yeah.
And then it's a lot harder to demonize GMOs, which is one of their favorite weapons, trying to fight against an entirely different societal division.
Interesting. Yeah, I'd never thought about that dynamic happening, but I think you might be right there. Nonetheless, I think you sort of mentioned field trials are an important countermeasure. There might be other countermeasures that we can sort of discuss. But the moral imperative is that we should be making progress on making these technologies safe enough and acceptable enough for use at, one, eliminating species, and then eventually maybe to improve the ecosystems around us in the distant future, right?
Yes, although I want to stop you because a lot of listeners are probably reacting to eliminating species with horror. So many folks want to optimize for biodiversity. I always advise my colleagues in conservation to be careful what you wish for because you might actually get it. If what you want to do is optimize for biodiversity, you want as many diverse species with different beautiful traits. I'm all in favor of that, remember? I want to rewrite the tapestry of life to be more humane and more beautiful and more interesting than it currently is. But most of them don't really have that as their real optimization target. They're trying to optimize for a return to some past ecosystem. Some state from before humans came in and mucked it up. And we absolutely do come in and muck it up and drive species extinct and change the ecology in ways that are not always good for the species there. So it's not that they don't have a point, but why choose that particular ecosystem with that particular time point as the thing to strive for? That's an open moral question. And if you want to actually say, oh, well, we're trying to optimize for biodiversity because that's what humans have destroyed, without specifying, no, the particular biodiversity that existed in the past, then I'm going to say, well, okay, I can use a gene drive to change the traits of this one species, I can speciate them, in fact. And I can give one half a different trait than the other half that they didn't have before. And we can do that again, as many times as you want. Adding new and more traits. That's biodiversity, right?
Yes.
So, and I think that actually has some lessons perhaps for folks in AI safety would probably seize on that and say, be careful what you wish for.
Yes.
Because there will be unexpected consequences that you have probably not thought through.
Yes.
But driving species extinct, well, with the screwworm, I think, frankly, that's the right thing to do. The screwworm wouldn't suffer from this, right? If you never exist, it's not a problem. And the way you'd be getting rid of them is either turning them all into males or ensuring that the females gradually become less and less fertile. You'd have to, it's not going to happen just with a handful released. You would need to release them throughout the landscape because otherwise you get this rock, paper, scissors dynamic where the local population goes extinct, gets invaded by a few viable screwworms without the gene drive, recolonizes, gets invaded by the gene drive, goes extinct again. And so the overall population drops to a tiny fraction of what it was before. But you don't actually get rid of them unless you deliberately drop gene drives, organisms in every relevant population that still lives, which is, in the screwworm's case, would not be that hard to do with drones and monitoring and so forth. But it'd have to be deliberate. But the best thing about the screwworm is you can actually freeze the larva and resurrect them, I mean, now we know decades later. So this is a species we can literally put it on ice, remove it from the wild, and it won't be extinct. We can bring it back whenever we want.
I see. That's an interesting concept. Just like, hey, all right, I see that you guys really like all species, but just this one, can we pause it for a while? And you tell me if you still want it back, right? I think this is a really good time to transition to these concerns around AI safety, particularly biohazard risk, particularly engineered viruses, gene drives that would do the same thing for humans. Talk to us a bit about your biggest fears.
I guess you can summarize this as, I am afraid that there is going to be some form of disaster arising from biotech that leads to a backlash that prevents us from using it to its fullest potential. I hope something really good happens, like getting rid of malaria, before something really bad happens that would make it much harder or impossible to do anything like that for a decade or more. A single tragic death in an ill-planned clinical trial set gene therapy back by a decade. Right now, we're seeing effects on science from politics that were at least fueled by suspicions over where COVID came from. Imagine how much worse it would be if that was deliberate. If, despite warnings, scientists deliberately made viruses worse that then were used as a weapon. I also suspect that one can make biological weapons that are rather worse than anything that nature does, because nature is optimizing for replication, not causing harm. That suggests that it's hard to engineer fitness in the wild better than nature. Obviously not impossible. Look at gene drive. Look at outside cases like mirror-bacteria. I think it can be done, but by default it's very difficult. In contrast, engineering something to cause more harm than natural things do, that's not something that nature is optimizing for. In fact, it's selecting against that. Viruses do not want to harm us so much that they change our behavior in ways that would prevent us from transmitting them. So this is a check on the virulence of natural pathogens. The nastiest ones, historically, things like the Black Death. Why was that so bad? Why is Yersinia pestis so deadly? And the answer seems to be that it's transmitted efficiently by fleas. What happens to an animal when it dies? What do the fleas do? They abandon it and go to seek to a new host. So because that transmission vector existed, there was little fitness cost to the bacterium to killing us. And so it could be transmitted airborne, yes, but it could also be transmitted from fleas. And so the cost to it of killing us was just much, much, much reduced. And that's in part why it was so lethal. If you look at rabbits, my existence proofs for, yes, even nature can come up with something that could really threaten us, could threaten civilization itself. Rabbits have myxomatosis and rabbit hemorrhagic disease virus, two separate agents that have eliminated more than 90% of living rabbits across entire continents for multiple continents. And for both of them, it's because they're also spread by various biting insects that can transmit it from a rabbit corpse to a living rabbit. And so there is selection pressure, or at least there is relaxed selection pressure for keeping the host alive.
I hear you. Okay. And I think this is touching on this idea that, yes, engineering a virus that optimizes for both having a high R0 and also having high lethality can be quite difficult, but as you said, not impossible. And now we have this sort of democratization of biotechnology and at least like intelligence, like the commoditization of intelligence through these LLMs. And then these LLMs are also being themselves commodified through open source LLMs that are only a few months behind the frontier. And this is scary, right? Because as much as sort of OpenAI and Anthropic do in terms of putting barriers in front of or sort of warnings and declining to sort of fulfill your request if the request is deemed to be of a biohazard sort of nature. Eventually, right, as intelligence gets commoditized, you have this offense winning. Like the destructive capacity of a bad actor becomes much more than the creative capacity of the world to sort of defend against it. And I wonder, like obviously this is something that you believe in. What can we do about this? What are you doing about this? And sort of how can our listeners help?
Well, I would have to qualify a few things about that.
Sure.
It is extremely difficult to accept that your own field might just be strictly offense dominant, such that there is almost nothing that your field can do to fix the problem that your field is creating. I have struggled with this for the last decade because gene drive caused me to think pretty hard about, is that offense or defense dominant? Turns out gene drive is unusually defense dominant because, as I mentioned, we built the counter, a gene drive that overwrites and changes back whatever was altered when we built the first one. And because of that, because the counter spreads exponentially just as well as the original, it doesn't really gain the benefits of exponential spread, which normally favors the attacker. But most of biology doesn't do that. Obviously, our COVID vaccines did not spread as fast as the virus did. In fact, it cost us 6 orders of magnitude more to create that vaccine and manufacture enough doses for all Americans, never mind the rest of the world, just for all Americans, than it would to make that virus via reverse genetics. Which is to say, if you discover a new pandemic-capable virus, and you share the genome sequence, and the reverse genetics protocol say already exists, and you say this virus is pandemic-capable, you're just giving an incredibly offense-dominant tool to everyone in the world. Because anyone can make and release that virus by infecting a handful of people and cause a pandemic for 1/1,000,000th of the cost of making a vaccine. And note that that vaccine will be much slower than the virus. That's just the definition of offense dominant. What if we develop biodesign tools that can predict which combinations of mutations in that virus will allow it to evade current population immunity and still transmit efficiently? Such that for every major epitope typically recognized by our neutralizing antibodies, because antibodies are the ones that block transmission, T cells mitigate symptoms, generalizing, antibodies block transmission. If you mutate all of the epitopes the neutralizing antibodies normally bind to, because again, combinatorial mutation, something that nature is not going to do, then anyone who can make variance sufficient to match the accuracy of your predictive tool. If your predictive tool says 1 in a million of its predictions will actually be good enough, then someone has to make 1 million different predicted variants. If it's 1 in 1,000, then they have to be able to make 1,000. If it's 1 in 10, then they have to make 10. It's not like the tool has to be able to perfectly predict, yes, this one and only this one will do it. Because no one in their right mind in synthetic biology assumes that their first design is going to work. This is the whole reason for screening and selections and directed evolution. You make a lot of designs, and natural selection in the case of a pandemic will favor the most transmissible ones. There's a lot of people who I respect who want the best for the world, who are developing these tools because obviously, if you can predict which mutations are going to escape your vaccine, then you can redesign your vaccine to make it harder to escape. And most of us don't think about potential misuse. Frankly, at all. It's unpleasant. I mean, we spend our lives trying to develop biological tools to make the world a better place. Especially folks who are interested in fighting infectious disease. The notion that someone would weaponize infectious disease, would use your own tools to leverage the thing you're trying to banish from the world as a weapon against others is just horrifying. You just don't want to contemplate it. The brain flinches away. But can you have a tool that will predict which combinations of mutations will escape current population immunity without having a tool that will tell you how to cause pandemics? And if you can cause one pandemic by escaping current population immunity, how many different combinations of different mutations of those major epitopes exist? Sequence space is so vast. There are almost certainly many solutions to that problem, each of which would require a new vaccine. So don't just think about the millionfold difference in cost. What if the adversary makes a bunch of them and 6 of them have the relevant fitness? All of them would require their own vaccine. What if they release them in stages? It's just not obvious to me that the capability to predict which viruses will cause pandemics in order to better design vaccines can at all be disentangled from the ability to cause pandemics at will.
This is depressing. Kevin, this is the last question. What's the good timeline here? How does humanity act over the next 5 years to prevent this this potential catastrophe, especially as these LLM tools are just commodifying, right?
Well, the hope lies in the fact that even though biology, I would say, is hardware insecure, evolution has not been selecting for defenses against a competent, malicious adversary intent on causing harm. Don't assume that any medical countermeasure can thwart a determined adversary who is competent. And AI tools will make all adversaries competent eventually because they're going to be better than we are. I don't know on what timeline, but eventually they will. But that requires people to be exposed, people to be infected. And all of the greatest threats are those that can spread on their own. Which is to say, our vulnerability comes not just from the fact that we're hardware insecure, but that we that evolution hooked up all of these insecure devices on a biological information superhighway in which various microorganisms constantly pass from hardware system to hardware system, that is, from each of our bodies to other bodies. This is something we can engineer the environment to just stop. So the solution to the pandemic problem is the solution to the infectious disease problem. Make the environment too hostile for microorganisms while remaining harmless to us. And there's multiple ways we can do this. In the long run, we want far UVC lights in all of the rooms, which are absorbed by proteins. We are multicellular, doesn't hurt us. But if you're a bacterium or a virus, too bad for you. You need those proteins. You can't afford to just have your top level of cells which don't replicate take damage. Doesn't hurt our eyes or skin as best we can tell. Obviously needs more safety testing than has currently been done, but looks super promising. Glycols, already in stage fog, already in most of the foods that we eat. Triethylene glycol and propylene glycol in the air can reduce the number of floating around bacteria and viruses by somewhere between 10 and 100-fold, right there. Generally recognized as safe compounds. And in an emergency, if we had enough really good personal protective equipment, at least elastomeric masks, one that you can check to make sure you have a seal because you literally press on it, and then if you can't breathe anymore, you know it's working. If we had enough of those for all of the workers who are responsible for producing and delivering food, water, energy, and law enforcement, and ideally healthcare too, then we can withstand any pandemic. Any pandemic. For long enough to make enough of those for everyone. So it's a race. We need to delay the disclosure of how to make the human equivalent of myxomatosis or rabbit hemorrhagic disease virus, and prevent access well enough or deter adversaries for long enough that we can build those non-biological countermeasures that will let us just air gap all of these hardware insecure biological systems. We already have virtually all of the technologies we need. We just need to actually use them. And the great tragedy is that after COVID, everyone wants to forget. We don't want to face it head on. We want to believe that our innovation in our field can solve this problem. And I just wouldn't bet on it. But it's true that other technologies in other fields can solve our problem. Biology is offense dominant within biology, most of it. But as soon as you institute spatial controls on biological information transfer, the problem goes away. We can do this.
Fascinating, Kevin. Well, I'll leave it there. It was great to have you on the podcast.